Assalamu alaikum everyone, this is Zainish Khan and today I will share you the procedure of gram staining. First, let me share you what key points you should remember when doing gram staining of bacteria. Gram positive bacteria stain purple, gram negative bacteria stain pink or red maybe. Also note shape and grouping of stained bacteria while doing microscopy. Let me share you some examples with the simple slides of identification by gram staining method. Gram-positive cocci, purplish pink, round shape. Examples are Staphylococcus and Streptococcus. You can see these microscopy slides of cocci bacteria. Gram-negative cocci, red in color, round shape, and they are very rare. Gram-positive bacilli, purple rods. Example is very common, Bacillus subtilis. You may also identify gram-negative bacilli, red rods. Example is very common, E. coli or Escherichia coli. Let's start our procedure. First, check out the requirements. For gram staining, you may need transparent glass slides, cover slip. It not only gives viewer better control over specimen but protects the microscope as well. The cover slip protects the ocular lens from damage by acting as a barrier. Acetone or 95% ethyl alcohol, crystal violet solution, grams iodine staining solution, Safranin solution, Cedarwood oil solution, some kind of pure culture or sampled of found colonies. Friends, I am professional microbiologist, but as like you all, I do mistakes, so apologies for that in advance. Here I am explaining you my experience that how I do gram staining. So let's get started. These are two cultures I have selected to show gram staining. One is S. aureus for gram-positive cocci in chains and another one is Bacillus specizini for gram-negative rods in chain. I will pick colonies from these plates to perform gram staining. I have picked these two slides and will mark with the code of organisms I already know, like SA for S. aureus and BS for Bacillus subtilis. I am also marking the smear place for my easiness. You can avoid this step. The glass slide is actually a transparent thing, so this marking will really helpful. For picking up the colonies, you can use wire loop or toothpick. And trust me, toothpick will really helpful if you are picking up the colonies carefully. Wear personal protective equipment whenever it's mandatory. In gram staining, it's not very necessary to wear gloves, but we should wear to avoid contamination and hazard-related issues. First, add distilled water drops on both slides to make smear easily. I am picking colonies of Bacillus subtilis with toothpick and S. aureus with the help of wire loop because I want to show both will work. I picked colony of Bacillus subtilis and now mix properly into the water. After adding the culture, I will discard this toothpick. Now let me show you how I pick colony aseptically with the help of wire loop. Cool it down first and then pick little colony and put into drop of water that we already placed on slide. Again sterilize your wire loop until it become red hot so you may not contaminate by this colony or culture. Let smear air dry or by blotting method. It will take 10 to 15 minutes. You avoid this distilled water drop step and directly use culture to make a smear with picking up very little colony, if you can. So my both slides are ready for gram staining. One is of S. aureus and one is for Bacillus subtilis. Let's start our staining procedure. Add drop of crystal violet on both slides to cover the smear for 1 minute. After waiting for 1 minute, wash the slides with water for few seconds. Now our next step is grams iodine. Treat with a few drops of grams iodine for 1 minute and again wash with water. And after washing, we decolorize it with 95% ethyl alcohol. So here we are waiting for 1 minute and now we are washing it with distilled water. After washing, we are using gram decolorizer that is actually 95% ethyl alcohol and then we wash it with water immediately because ethyl alcohol can damage the smear. if we put it too long. Let the smear dry by blotting. With the help of Safranin solution, I will do counterstain for 30 seconds. Again, wash with water. If your slides are wet, dry it with lint-free cloth or tissue first, then blot dry. Now add few drops of cedarwood oil. Now these slides are ready to be examined under microscope at 100x. Hope you liked this gram staining video in detail. Thanks for watching. Take care.